UNAM.
Okay, well, thanks very much. First of all, is this loud enough? Can everybody hear me? Okay. Well, we are very happy to be here. We are a recently formed team, so rather than speak about results, we will talk about ongoing work. And basically, I'm supposed to give you just a very small introduction about who we are and the people that integrate our team. We are, well, two of the leading institutions in Mexico. IPN is a technological institute, and UNAM is the Nacional-Universidad of Mexico. We are four institutions, basically biologists from UPIBI. I think it's not worth telling you the names in Spanish, so I'll just read the sort of translations. Biotechnology unit and the School of Computational Sciences. That's from the Institute of Technology in Mexico. I'm from the National-University, the School of Science, and the Applied Math Institute. So, first of all, I think that you have to realize that all these institutions are in Mexico City, and well, communicating in Mexico City is not an easy job. That was a major problem, and we are still trying to overcome several starting problems. But anyway, we have a definite proposal to make, and in the next few minutes we will divide the presentation in three. First, the biological setting and some sort of introductory remarks from the biological point of view. Then I'll talk a little bit more about the modeling part and what we are trying actually to do with our system, and finally we will talk about the connection with computing. So,
well, does everybody hear me? Yep. Okay. In UPIBIS we have like the biological approach, and we will try to reproduce an experiment carried out by Michael B. Elowitz. So we will try to generate an genetic signaling cascade, so we can get a expression repression green fluorescence protein system. So the function of this switch is based on the constitutive expression of the repressor protein, which inhibits and generates a negative feedback loop of the green fluorescence protein, and it's another promoter. So we aim to control this inhibition caused by it by the addition of ATC, external ATC. So we saw that what we try to get is that when ATC is added, we can get a turn-off state of the green fluorescence protein of the TET repressor inhibition over the green fluorescence protein. So how will we get this? We have two feasible ways to get it. So we have chosen, we will make the two experiments. In one we will get everything into only one plasmid. So with this it will be easier to get it into the cellular system, and the other part of the experiment will be like to make both of the genes into different plasmids, each one, so we will be able to introduce it easier at into the cellular system. Well, with this we aim to we aim to improve our molecular genetic techniques, as well as we improve our biobricks management, manipulation, because we have just been formed, so that's like the biological approach. So with this we will implement computational and mathematical approach, non-conventional one, so we can get a better systems accuracy prediction, and well, we improve as well on circuitry function. So computational and mathematical approach will be just explained now. Bye.
So let me first mention that this idea of implementing a very, very simple circuit was made for two reasons. First of all, because we wanted to gain some experimental know-how, but the second was because we were working on Arabidopsis thaliana, which is probably known to many of you. It is a very small flower, and we had identified particular genetic subnetworks, specifically one that is responsible for the formation of hairs in leaves and root. And we came up with a mathematical model that incorporates all the inhibitory actions in one and all the activating actions in another, what we call the activating complex. So we suggested a reaction diffusion type of partial differential equations in order to generate a Turing pattern. And so we got this published, but I'm a mathematician, and we've been receiving many comments from biologists opposing very, very strongly to the fact that reaction diffusion systems and Turing patterns actually do appear in nature. So our proposal, a specific proposal, is to build a very simple system of the activator, inhibitor system, like the Gier and Meinhardt typical construction, but based on a genetic circuit that can actually reproduce Turing patterns. We've seen similar or related works from other teams, particularly I think the ETH group in Zurich, and other groups that are also interested in Turing patterns, more approaching the problem via chemotactical interactions and other things. But specifically, we saw that there are interest. For us, it is a very interesting problem to see if actually, as it has been proved in chemical reactions where Turing patterns have been obtained without any doubt, these can be also obtained in genetic circuits with a very simple proposal. So we already have a mathematical methodology. We have some experience, let's say, converting the logical rules given by the genetic network, then building a discrete dynamical system, and afterwards from this dynamical system, which is basically what you might regard as a cellular automaton, you can construct a set of ordinary differential equations, put some noise in there and get a stochastic differential equation, and from that write what we call the Fokker-Planck equation, which is the equation for the transition probability. If someone is interested in this, we have some work done already, but the basic aim is I mean, the big picture is more or less that. And with that, we want to implement some non-standard computation. And about that, Rosaura is going to, she's more comfortable reading what she has to say, so she's going to read her part.
Thank you. Well, we are talk about the modeling proposal by SCOM's contribution. Developing models to describe inside the cell metabolic event the cellular automata processes. We have a definition. We talk about the cellular automata are discrete or continuous dynamical system studied in computability theory, mathematics, physics, chemistry, artificial life, and biology. A cellular automaton system consists on an infinite regular grid of cells. Each cell takes a defined state from a finite set of states. Cellular automata can evolve in 1, 2, 3, or any number of dimensions over time. How does a cellular automaton function? Well, the important element is the local function. The local function defines the relationship between the central cell and its neighbors, so that a given neighborhood determines the evolution of the cell within the finite set of states. All possible relations produce the specific local function. Thus, we can calculate the history of the evolution. Each time or generation represents a global state of our system. There we can see the dynamics of the function. In this way, all cells are updated using the local function in parallel each time. Cellular automata are often simulated on a finite grid rather than an infinite one, due to the fixed limit of available memory. We focus on two-dimensional binary state cellular automata, where every cell can take two states, 0, dead, or 1, alive, and updates each state depending on the sum of states of its 8 closest neighbors as follows. In particular, we studied a special complex cellular automaton system with a local function called diffusion rule. This local function works in the following way. Cell in state 0 takes state 1 if there are exactly 2 neighbors in state 1, otherwise the cell remains in state 0. Cells in state 1 remain in state 1 if they have exactly 7 neighbors in state 1, otherwise the cell switches to state 0. Complex cellular automaton governed by such local transition rule exhibit reaction diffusion like patterns, stationary or mobile particles, particle guns, and chaotic behavior. We have 2 links that we are explain our work. I have copies if any team is interested in this. And we using the diffusion rule, we can generate dynamical patterns of cellular reaction, like turn on off light with alive or dead cells that show luminescence. Examples include fluorescence, bioluminescence, and phosphorescence. It's necessary to construct the initial basic reaction. The basic pattern is produced with 24 diagonal alive cells formation. In each generation, the pattern is transformed with perfect control because our system doesn't grow forever or disappears. The population has an equilibrium of number of alive cells. The simulation shows this phenomenon. The cells alive are represented in yellow like an activator, and the dead cells represented in black color like an inhibitor. The luminescence phenomenon was obtained by the evolution of this initial pattern with diffusion rule over thousands of cells. The process takes several stages where the luminescence phenomenon may be reached. Finally, our construction after 24 generations is periodic, oscillating with blinking patterns of period 2, apparently forever. In the last, we have a long-term research, our Team Mexico. We have think about creation of new biobricks, virtual reality, and biological system, simulation of biological events through cellular automata, Turing patterns generated by simple genetic circuits, and non-conventional computed applications. And we have a reference in this page. This is our presentation. The team is formed by these people. And thank you.
Are there any questions?
Can you connect what you're going to do with the simulation to the actual experiment? What's the first type of experiment you might do?
We can do simulate a fluorescence in a biological system, and we can, using the diffusion rule, simulate in a computer experiment. We can connect these ideas using a cellular automata.
If I may just make a little bit of that question. The first simple experiment is to build an activator inhibitor genetic circuit, and that will be really the start. And then we have to see if it's really feasible to the rules.
So just if I may follow up, is it possible to make a system, whatever its rules are in the biological system, and then fit the cellular automata to derive what rule the cell is using?
Well, that's exactly our long run goal. That's the difficult part.
We have some copies about this work if they are interested in this.
One of the features of reaction diffusion systems, of course, is the interconnectivity of the system, the diffusion of information from one cell to another. In the biological system, have you got any ideas for how you would implement that?
Biological systems, we are,
Yeah, well, that's why biologists don't work in Turing patterns, because they claim that the inter-cellular signaling and other things are not diffusive. So what we claim is that at least as a first approximation, you don't have to implement that. I mean, you have some sort of first neighbor interaction, and that can be modeled effectively by diffusion.
Yeah, it's just that in the Arabidopsis system that you showed, the MIP-D proteins, the small.
For instance, yes,
Some
of them diffuse. Yeah.
Any others?
So about the composition of the team, what was the balance between biologists and computer scientists?